Welcome, everyone, to Catalio Conversations, where we talk to founders and CEOs across innovative healthcare and AI who are building the next generation of disruptors in the space, from drug discovery to autonomous labs to clinical intelligence and beyond. I'm your host, Martha Petrocheilos, and with me today is Sri Kosuri, the co-founder and CEO of Octant, a clinical stage drug discovery company using synthetic biology, high-throughput chemistry, and AI to discover small molecule drugs for complex diseases by measuring how drugs behave in human cells, both for efficacy and potential side effects. Sri, welcome.
Thank you for having me.
Of course. So glad to have you. So let's kick it right off and go back to the beginning. So before Octant, you had a very established academic career at UCLA, and you had already helped build a couple companies. And then a discovery came out of your lab that was too important to just set aside. Tell us a little more about that turning point and what gave you that conviction, really, to leave a very secure path and go pursue Octant instead.
Thank you. It was the culmination of a bunch of things that we were trying to do in the lab that was starting to come together broadly about a series of technologies around DNA synthesis and editing, but really also doing lots of measurements in cells at the same time using next-gen sequencing. And those coming together really drove a class of experiments that I was blown away by and thought it would have broad impact on drug discovery. And the only way to figure that out was to start a company to try to build new drugs that other people couldn't.
You chose a more direct path to impact and wanting to reach patients faster.
Yeah, I mean, if you want to build drugs and you want to build drugs in a new way, you kind of have to go do it. It's not really possible in academia. I like building new things. I've always have in my career, and it actually seemed like a lot of fun.
One of your lab's breakthroughs, of course, was finding a way to make building genes far less expensive, which is an advance that you could easily have sold to other scientists, right? Like a standalone business. Instead, you took a harder road and decided to develop medicines yourself. What made that worth it for you?
We did actually start a DNA synthesis company. It got acquired later while in Boston. But I did that in more traditional paths, like the tool companies where I sat on scientific advisory boards and helped guide those companies from a large distance. This was different in the sense that it felt like where my career was headed. I wanted to really see if this could lead to different ways of building drugs in an exciting area, in small molecules that did things starting to emerge, but we didn't really have an engineering platform to build those kinds of drugs in the past.
So the tools that you had built were necessary to now start your own company and develop drugs in a very novel way. How exciting, right?
Yeah. I mean, to be fair, it was, naive. You learn a lot, but, I think that's how things get done. That's how you do new stuff, right?
Was pressure by investors or otherwise early on to stay a tools or platform company just because it was less risky, perhaps a little cleaner?
Our largest investors have always been supportive of what we wanted to do and tried to magnify that. We wanted to build drugs from the very start. What we got to learn is what types of drugs and what areas and what can you build. That took a lot more evolution, but we wanted to build a drug company from the start.
That decision also meant deciding where the platform would matter most, right? Octant, of course, began with the idea of designing drugs that do multiple jobs at once. Today, your focus has shifted to correctors, to be more specific, which are drugs that treat diseases where the body builds a protein incorrectly. And this includes forms of inherited blindness and certain genetic disorders. What led you to concentrate there?
This goes back to the lessons of what we were naive about and what we weren't, right? We had started with this idea in dirty drugs, in a class of receptors called GPCRs, where there's a lot of polypharmacology, where a small molecule can hit many things, and that's essential for their function, and bias, which is multiple pathways off the same target. We switched largely because we began to realize that if we wanted to build scale and our own commercial drugs, it's really hard to do that in a large, complex disease area. Your only option really is to transact the asset where it value inflects. And we were trying to build a company that can build scale over time. What we saw was small molecule correctors that we landed on are more protectable in the sense that they're really hard to build. No one else has really good ways to build them. And when you build those drugs in rare disease, patients come to you. It gives you time to build the commercial infrastructure infrastructure to really deliver those drugs at scale and benefit from cash flows to continue to reinvest and build that future. We wanted to build a next-gen pharma. That's our ambition. And we thought that rare disease was the right way to do that. We're trying to build very pan-mutant correctors that rescue hundreds of mutations across genes so that we could have one drug that can serve a large proportion of the patient.
So it sounds like the shift was towards a problem that the rest of the industry had largely given up on in many ways.
Not even approached.
Not even approached, exactly, kind of steering away from. And of course, correctors really fit your platform really well, right, Sri? They've traditionally been very hard for one target screening. So that was another thing that probably just fit well, right?
Yeah. Very early on, we recruited Mark Mirko to the board and then to the company. He was one of the founding CTO at Vertex, a large biotech. It's $100 billion plus market cap. Its major drug is a set of small molecules that rescue mutations in a gene called CFTR that leads to cystic fibrosis. And today it treats about 90% of the patients with mutations, sells about $10 billion a year, but it also has no competition, right? And it's because correctors are really hard to make. And the reason is because an unfolded mutated protein has no structure. So structure-based drug design goes away. You can't purify it because it's degraded. So biochemistry goes away. You have to run chemistry in cells, and it's a very empirical process that takes tons and tons and tons of iteration. Our platform is really good at that, and so we thought we could do this across a broader range of diseases.
And talking about the high degree of complexity that is involved with correctors specifically and the high levels of iteration, was there something that you got wrong in the early days? It could be science or the business side of it. And how did that change how you run Octant today?
You're kind of building the plane as you're falling off the cliff.
As you're driving.
Yeah. All of those lessons you learn and missteps that you make, you just learn each time. And one of the beautiful things for us has been focusing on a mechanism like correctors, is those learnings have repeated across programs over time in ways that made things a lot easier, essentially.
I love this framing. Did you ever have pressure to keep the platform more broad than it was, as opposed to sharpen and specialize the company as time went on?
Yes, absolutely. Our lead program, Rhodopsin, in retinitis pigmentosa, it's really scary to go after a first-in-disease market for a startup and a first-in-class, first-in-disease market, right? But now that we've kind of gotten over that hump, it's wonderful. It's a huge deal, and we're not fast-following someone else and don't have to sell the asset, something we can commercialize ourselves. Our second program in Fabry disease is actually a much, much better corrector than something on the market that is selling. We think of our portfolio as a way to spread those risks in different ways. What should our second program be? What should our third program be? Something that's most investors in the biotech space probably feel right now. When you get to our stage in a company, no one wants to pay for a platform. They want to pay for those clinical readouts.
The data. We'll talk about this later.
The data. But what we've done as a company is do partnerships in ways that are not core to what we're doing, but pay for the platform carrying costs over time. What alleviated that pressure over time for us is that our platform runs fairly net neutral, and that lets us just focus our equity investments into the clinical readout.
And that brings us to the machine that you're building and that you've built. Of course, traditional drug discovery is remarkably slow because it tests drugs against one target at a time, although a single disease often involves many targets, right? So your system does something different. It tests thousands of drugs every week against many disease targets all at once. What actually is happening inside the platform when you run one of these experiments, and why is it so unique?
Our platform, as you mentioned, does a lot of high-throughput biology and chemistry and AI and ML and automation. The best way to understand it is the parts of the drug discovery process. There's a lot of ways to kill a protein's function. You can kill its active site. You can misfold it. You can send it to the wrong place in the cell. Our platform on the biology side, what's really exciting about it is because we can do experiments at scale, we can build every possible mutation that might ever exist in patients to this gene, read out the different activities in cells, and then understand with genetic data sets that are widely available now, what percentage of these patients actually have misfolded variant? And it turns out to be a ton of them. And so we thought if we could build a corrector that treats a bunch of those mutations, it would be a valuable drug. Even that information is super valuable to us, right? Because we're trying to go after low biology risk problems. We're trying to treat the underlying mutation that causes disease and the mechanism that causes the disease. And then it's like, as you were saying, what's interesting about cystic fibrosis and what Vertex did is that it's a pretty special rare disease in that it's dominated by a single mutation in white European populations. There's so many people with cystic fibrosis, but they all have the same mutation. So Vertex really had to fix that one mutation. Most rare diseases are not like that. There's hundreds of mutations that lead to disease. And so you have to build a pan-mutant corrector, or as many mutations as you can, to build something that's both clinically and developmentally viable, but also commercially viable. You can't build a new small molecule for every person. You can't build a new drug for every single mutation. We are trying to build pan-variant correctors, and that's a multi-parameter optimization problem, right? And so what we do is in a single well, we can do hundreds of assays. We can run tens of thousands of wells at a time. What our high-throughput chemistry platform does is basically we get a hit on one of them. What we can do is build a bunch of analogs of those molecules, about 5 to 10,000 at a time per program. We do that every week. And so it lets us evolve these molecules to have broader mutational coverage. For the rhodopsin program, the first programs was about 5,000 compounds a week, every week for a year. So 250,000 analogs over the course of that year to get to our lead compounds. Today, we're doing something like 100 to 200,000 compounds a month across 2 to 3 programs. So we basically hammer this thing. That requires a ton of automation, a ton of software, and a lot of ML and AI to understand that data, decide what the next experiments are going to be, and then execute those experiments.
Those are incredible numbers. I hope people watching realize how out of sync this is with how things would be done in a traditional lab. So to put this in perspective, how long would that same volume of testing take a traditional lab?
There are campaigns like Vertex took 10 to 15 years to build their drugs, and they used hundreds of chemists across a decade. They built about 75,000 compounds in much slower iteration cycles. These kinds of problems are hard to solve.
Where does the biggest speed advantage come from? Is it the barcoding? Is it the automation? Is it the data feedback loop? What do you think?
Is it lots of pain and suffering over 8 years of building that?
It's the emotional suffering, actually.
It's like building a factory, right? It takes years of learning where the bottlenecks are, fixing that bottleneck, finding the next bottleneck. But on the chemistry side, we run a process that's known as direct to biology. We're never purifying compounds individually. We're doing raw chemical synthesis in a sub-microliter droplet, and we directly dose these cells. That requires this collaboration between the chemistry and biology that we have to work on chemical reactions that are non-toxic enough for cells to read out on them. And on the biology side, you have to work on building the set of multiplex controls that can look at these differential readouts so it can tolerate a little bit of messiness. And then that requires a whole engineering mindset around turning this into a crank that you can turn every day and every week. And then obviously the computational infrastructure that sits on that so you can understand that much data week to week and then decide on what's next.
So it's multimodal, it sounds like, multifunctional. It doesn't really stem from one single science, right? There's the chemistry, there's the biology, there's the engineering, there's the business side, of course, that we talked about. It is truly multimodal, very interesting. And because you test inside living human cells rather than isolated proteins in a test tube like many others do, a single run shows how a drug actually behaves in a realistic living system. It doesn't just show whether it sticks to the protein, right? So why is testing in real human cells across many targets at once such an advantage over the traditional approach?
Well, the traditional ways to build small molecules are to do biochemical tests or structure-based drug design. That won't work for correctors, right? You can't purify a mutated protein. There is no structure. The processes that govern an unfolded protein and what it has to go through to get fixed is a set of processes in cells such as the unfolded protein response, the protein quality control machinery, post-translational modification machinery. These are hundreds of proteins in humans that coordinate actions that can't be modeled through AI, it can't be modeled biochemically. You just have to use cells. Those cellular pathways are found in every cell in your body. And so it's about doing things across that machinery and trying to find compounds that help guide those proteins through that to reestablish function.
So you're seeing problems and opportunities that like a simplified test in a tube would simply miss entirely.
Yeah, and the other thing that's really interesting in this is binding separates from function. What's really interesting about Vertex's correctors is they're not super strong binders. That's not how these drugs work. And there's a whole class of emerging drugs. Correctors are one of them, induced proximity, expression modulators, splicing modulators. These are the future, we think, of small molecule drugs because they can do things that we never thought small molecules could do. But they're going to require a whole different way to think about building them in reliable ways.
How often, I wonder, does a drug that looks perfect on paper inside an isolated protein actually falls apart once it's inside a living cell?
Yeah, I mean, there's more than the on-target activity. There's all the other ways that you need to optimize. So it's not just does it get to the right place and does it do the thing across mutations, but does it get to the back of the eye? Does it get degraded in the liver the moment that you take it? Is it orally bioavailable? That is a super hard challenge too. What's been really exciting is bringing those kinds of data sets and assays also to the platform. And we've been doing that over the last many years. It is part and parcel with why these drugs are difficult to make.
And of course, you're using AI now to catch a drug's likely side effects very early before it ever even reaches a patient, as opposed to discovering them later, where the failures are a lot more costly. Why has the industry, you think, settled with finding drug side effects so late in the process? And why are you confident that you can catch them sooner?
Well, the industry hasn't settled on this. It's super hard. We're running a trial. It's an experiment, and we still might get surprised. We are trying to build models based on our data. We're also open sourcing a lot of this data, not for our compounds that we're commercially interested in, through a set of competitions and an organization called AdMet that we helped start, where we've been running blinded competitions around very large data sets around safety and toxicity, and let the world come and compete against them. Because we are one of the best at producing that data, we're happy if someone wants to sell us a model that does this. What we find is actually all these models so far are pretty much similar. And having the local data for your compound is actually super important. We haven't had our AlphaFold moment on small molecules. One of the things we're doing there is trying to see where's the field at? Where are we at? And in the meantime, we also are building, and because we have some of the world's cheapest ways to produce those data sets for our local chemistry. And so that's kind of how we've been navigating that space.
So the goal is not just to predict toxicity, right? But to make safety part of the architecture of the drug from the very beginning as part of the design.
Just a question of how many shots on goal do you have and how good are your models at guiding those?
Does it change the economics of drug discovery, do you think, in addition to the science?
I think small molecules have an inherent disadvantage. Part of the advantage of a small molecule is it can get to every cell in your body. It can interact with every protein in the cell. That's also its disadvantage. It's idiosyncratic safety, whereas an antibody tends to be safe unless there's on-target toxicity. So if we can solve the off-target toxicity problem in cells, we're trying to do that, then it becomes much more reliable, like an antibody. And that is a very powerful thing for small molecules if that can occur.
Truly revolutionary. You've earned confidence from very different quarters, right? Venture capital leaders, and of course, a Gates Foundation grant for cancer, your partnership with Bristol Myers Squibb. When a major pharma company decides whether to bet on you, what's the hardest thing that you still have to prove to them?
It depends on the partner, but if you think about classic pharma BD, that has not worked for us. No one's out there like, go find me the best corrector. We're not in a classic bucket.
It's such a unique drug.
Exactly. And we have a unique platform. Like there's not a ton of competitors to what Octant has built in this combined biology chemistry platform. What we end up doing is talking to R&D leaders. And if we can convince an R&D leader that this is a super interesting idea, they don't have any other option to partner with other people. We've been very lucky that Bristol Myers Squibb took a very early bet in the company, invested along with Catalio. It was around our very unique capabilities around building all possible mutations to a target. They had TYK2. They were building and developing a drug for that. And we were using it to interpret human genetic data sets. And we were the only game in town for companies that could do that. And so that helps. And then we build trust. And now we have a drug discovery collaboration with them in neurodegeneration around building a drug. It's finding some unique capabilities that someone wants and then building trust on execution to be able to do that again and again.
And I think also the ultimate proof point is just showing that the molecules behave better in the real world.
100%, right? Now BMS trusts that we can build correctors. We're working on a corrector project with them because we have a drug in the clinic that no one's ever seen before.
Nearly everyone in your field now calls themselves AI powered, right? And your view is different. You think the real bottleneck is not AI, it's having enough high quality data to feed it, which is why you build the lab first. Does the rest of the field eventually come around to your view, or does the AI hype keep winning?
The idea that data matters more than algorithm is there. We haven't seen some AlphaFold jump with AI ML models. It's really still in the data. If you have really good data, and it's not just any kind of data, part of it is picking the right assays, understanding the limitations of those assays, and then being able to run them at scale. And that is an art still. And we're very, very good at doing that. That's been our advantage. I think a lot of people think of AI as making things faster. Oh, I just have to make one antibody and zero shot it, and I'm going to have a drug or one small molecule. My hot take there is I think that's going to be less valuable over time. You have to build drugs that no one else can build and copy. You just have to be able to do that.
That's a unique selling point, right? I mean, the model needs a data engine. Otherwise, you run the risk that AI is just pattern matching on insufficient data.
That's exactly right. The AI needs a data engine. And B, you have to work on problems that require massive numbers of iterations. It's just a super hard problem. You need both of those things and combine them. A little bit more of a hot take too is I don't think enterprise value is going to accrue to foundation model companies. It's going to accrue to companies that learn how to do the drug discovery process more agentically through these loops.
That is the future. I think the drug company of the future, and that's my next question, very much looks like one where both AI and the lab work together in tandem.
Yeah, a lot of what we're trying to build now is how do we build the AI drug hunter, not the AI drug, the AI drug hunter. Like I have these 5 series. I should pare that down to 3 because those 3 are better than those 2. And like, that's where we should focus. Oh, and this assay is looking weird now that we built a better compound. And so we have to go back and do the assay development. In order to get that decision-making layer, you need the execution layer on the platform, but you also need this knowledge base on the program record. Andre Carpenthy calls it the auto research loop.
That is a very hopeful view. Looking 5 to 10 years ahead, looking out into the future, if Octant works the way that you hope that it does, what changes for a patient with a disease that a doctor can't treat today? What does that future look like?
Man, that would be an amazing future. Even our first drug, if that works, I will be really excited because that drug won't be produced unless Octant produces it. This is an autosomal dominant disease where you are largely blind and your children are going blind year after year. What would you not do to have a pill that solves that? There's hundreds of these diseases that we can go after, and everything from rare diseases like we've talked about, but we're also working on P53 correctors, the most mutated gene in cancer, or neurodegeneration, and many of the biggest unmet needs, principled bets, trying to build stuff that no one else is going to build and take the chances to see if they work. It's such a wonderful industry and just very proud to be a part of it.
You should be. What has to go right, do you think, between now and then for that future to actually arrive?
God, well, the simplest thing is we have to make money. We have to have successful drugs that are very valuable and drive cash flows that let us take more bets. That's what a drug company is.
It's a beautiful vision, Sri. It's a world where the undruggable, right, is not a permanent label.
Yeah, and I think it's only going to get more exciting. The amount of science that's changing and what we think small molecules can do, massively changing. The industry broadly is in a much better scientific foundation than we ever been before to build drugs, and it'll lead to a lot of change.
I love it. I think that's a perfect segue onto my quick fires. I have a couple for you. So let's go. One myth about AI and drug discovery that you'd like to kill in one sentence.
I think the value of AI to drug discovery is going to be largely driven by the same value that AI will provide to all businesses, not in particular focused foundation models.
Third hot take of the day. One bet you're making about how drugs will get discovered differently in the next 5 years.
We're making that bet at Octant, which is the era of target-based drug discovery as we thought about it is largely going to become commoditized. And these weird things like Revolution Medicine's wonderful new drug and induced proximity or correctors or expression modulators or just weird new drugs are going to be the dominant paradigm over time.
One thing about how the pharma industry works today that makes you think there has to be a better way.
I would love more founder-driven pharmas. I think Regeneron is probably the one that's only achieved scale, but there's something that comes from having a founder still at the helm that I think lets you take big bets.
They understand the complexities and have the vision.
Yeah, there's a power that a founder has that a CEO often doesn't. There are really great CEOs that are really good at doing this, but if you look at the top of the S&P 500, it's changed a ton over the last 30 years. That's not been true of pharma.
Sri, thank you so much for being here with me today. I really appreciate it. It was so great getting to talk to you and unpacking this. What's so incredible about Octant is not just the technology, but also, I think mostly, if anything, the ambition behind it. You've really tackled those undruggable diseases that most of the industry has steered away from for a very long time, and testing them in living human cells against multiple targets, getting to see how they really behave in a living system, getting a very good view of side effects as well as efficacy. I think it's so incredible and unique. Thank you so much for what you're building. It's incredible.
Thank you for having me on. And also thank you to Catalio. One thing George does for us is push us to be ambitious. That's been really great.
I am sure. Thank you so much, Sri. And thank you all for tuning into Catalio Conversations. If you enjoyed today's episode, please share, like, comment, and subscribe. And please stay with us for more conversations with the founders and CEOs who are changing the future of innovative healthcare and AI. See you next time.